This is C.B. Matthews, sir. I mean, he is our head of the department.
Yes.
Hello.
Hello, Professor
Hello.
Matthew.
Hello, Dr. Deepak.
So yeah, Jimmy. Yeah, I don't know whether you know. Jimmy told you we were classmates.
Yeah, I know. He used to share.
Yes.
I have given him your 11-page CV.
അയ്യോ പറഞ്ഞ കൊളവാക്കിയത് വേണ്ട അല്ലെങ്കിൽ ഒരു ഞാൻ ജസ്റ്റ് ഒക്കെ സെൻഡ് ചെയ്തു. That was the one that I had.
No, that's good. That's good. I mean, yeah, that's very nice. Yeah.
Now the whole job is entrusted to Jimmy only.
Okay.
He was not ready. He was not ready to share anything with me. He was the whole. I can say.
No. He was. I sent him the CV. Yes, I sent him the CV.
Anyway, so how are you guys over there? Everything good?
Yeah, it's going on. I mean, not. And a lot of students have shown interest. I mean, students from mathematics.
Okay.
Life sciences.
We will
and chemistry and physics. They all want to hear about this stuff.
I hope I can do justice to all these guys, but we will see.
Sure. I mean, Deepak got the best teacher award in IISc. Now he's telling that he can do justice or not.
No, we will see.
This is one of the most difficult awards. I mean, very prestigious award that he has got. So maybe I will again.
Yeah. When you want to start, we can start. No problem. I'm kind of
yeah. Maybe now you. I mean, let me ask him, Deepak. Sibi.
പറഞ്ഞോ സാറേ.
നമ്മള് ആന്തം പ്ലേ ചെയ്തിട്ടല്ലേ തുടങ്ങുന്നത്?
ആന്തം പ്ലേ ആണോ?
ചെയ്തോ അതിൽ തുടങ്ങാം. Deepak, maybe Sibi needs to access sharing, so you may have to get out, and then maybe later on upload.
Yes. I am already out.
Great.
Call the principal.
Yeah, I will call him.
സാറേ സ്റ്റാർട്ട് ചെയ്തോട്ടെ? സാറേ സ്റ്റാർട്ട് ചെയ്യുമ്പോൾ പറയണേ.
സ്റ്റാർട്ട് ചെയ്യാമോ?
സ്റ്റാർട്ട് ചെയ്തോട്ടെ സാറേ?
Principal.
സാറേ മ്യൂട്ടാണ് സാറിന്റെ.
Jimmy, you're mute.
Okay. Now first we will have the college anthem, and then
Sure.
So let me see if the father is.
പ്രിൻസിപ്പലിനെ വിളിച്ചു സാറേ. പ്രിൻസിപ്പലിനെ വിളിച്ചു.
സാറെ വിളിച്ചു. അച്ഛൻ ട്രൈ ചെയ്തോണ്ടിരിക്കുകയാണ്. ഒരു ഇഷ്യൂ ഉണ്ട്, ചെറിയ ഇഷ്യൂ ഉണ്ട്. ക്യാമറ ഓൺ ആവുന്നില്ല. അച്ഛനെ കാണാൻ പറ്റുന്നുണ്ട് നമ്മളെയൊക്കെ, പക്ഷെ ഓഡിയോയും വീഡിയോയും ശരിയാവുന്നില്ല.
അപ്പോ
ട്രൈ. Let's wait for 2 more minutes and then we may start.
Jimmy, I'm there.
Father, I can see you and hear you.
Yeah, because I took the wrong channel. I clicked the YouTube link. That's why.
Okay.
Okay.
Right. So, Sivi, can we start? Sibi, can we start?
Stopping by the shimmering shores of time, musing on the timeless truth sublime. Seek aside a gleam on flaming wings. Sacred heart, the soul enchanted sings. Blessed are we on this praising shore, blissfully gleaning pearls of lore. Pilgrims on these pastures green, seeking joy, hinterlands unseen. Sacred heart, a ball of light, shine on bright, a bright heart, sacred heart, shrine of light, shine your beams, make us one at heart.
Good evening, everyone. Invite our head of the department, Sibi sir, for the welcome speech.
Am I audible?
Yes, you are audible, very much audible.
Respected Principal Reverend Dr. Prashant Palakapalli, Honorable Chief Guest Dr. Deepak Nair, Program Coordinator Dr. Pastin, my dear students from SH and all other colleges. A warm greeting from Sacred College, Devara. This session is one of the rarest, being, as you know, it is a targeted one for first year students, science students only. At the onset, I would like to congratulate Dr. Jimmy for initiating such an idea, wonderful idea. മലയാളത്തിൽ ചൊല്ലി നിങ്ങൾ കേട്ടിട്ടുണ്ടാവും, കൊച്ചി പഴയ കൊച്ചിയല്ല. അതുപോലെ the science is not the old science which we get in separate packets like physics, chemistry, maths, biology in a compartmentalized form. Technology has changed everything. Now it is time for integration. Many of you might have read an article on the famous speech made by the famous scientist Richard Feynman. And the name of the talk is There is Plenty of Room at the Bottom. Made in 1959, December 29, at the annual American Physical Society meeting. He has raised some very bold unanswered questions at that time. Why can't the encyclopedia, the full volumes, can be put on the head of a pin? Why can't we have tiny motors using atoms? So, lot of such questions are raised by him. He is also considering the possibility of manipulating individual atoms as a more powerful form of synthetic chemistry to make more sophisticated materials. So, how is this possible? This is possible by high resolution microscopes and lasers. I hope this talk will give you an insight into all these aspects. Now we have our principal with us, Reverend Dr. Prashant Palakapalli. He is very much keen on science and technology, and he has a passion for science, though he is a social scientist. I am very happy to welcome you, Father, to this function. Then we have our resource person, is Dr. Deepak Nair from IIST Bangalore. He's an eminent scientist in neurosciences. Jimmy sir will be giving an elaborate description of what he is, being his classmate. We are very happy to have you as resource person for inspiring the budding scientists of the next era. Welcome you, sir. Then, my dear students from SH and other educational institutions, you are the people who are going to be counted for the technological advances and torch bearers to the next century. A hearty welcome to you all. Also, I request you to have an active participation and interaction during the coming session. Once again, I welcome you all. Thank you. Over to Jimmy sir.
Yeah. Invite our principal, Reverend Dr. Father Prasad, for the presidential address.
My dear everyone, Dr. Sibi, Dr. Deepak, and Dr. Jimmy, other colleagues, and my dear young friends. I feel really happy and also really jealous about this whole thing because I look back and see that this is no more possible for us. And yeah, we say that everything is possible and all that, but practically saying, I know that this will not be possible. And I see that it's a great, great opportunity of being given. And I'm retiring this year, and next year I have some other tasks already assigned. So I don't think any more science education will be possible in this manner. Maybe I will get to know some bit of science. And I record my small introduction to physics and chemistry and all this stuff in school days. Afterwards, I never had the opportunity to do that till I took charge as the principal of this college. So I deeply regret that I have missed out all this fun, all that fun, learning science and grappling with the realities around us and getting into the intricacies of life and the phenomena around us. So I believe we are getting a fabulous opportunity. I hope that at least you may not realize that right now. Later on, only you might realize. But my advice at this stage, I have started giving advices, though it's not really good to give advices. So, but still, I suggest that all of you take this very seriously. In the sense, take it as fun. I hope this will be really fun learning things and trying to understand things from people who are experienced in the field. And I hope that you will definitely make use of this opportunity to the best possible and demand more. We will be only too happy to oblige you by creating such opportunities further. Only thing is that often the demands are for holidays, often the demands are for postponing exams and labs, demands are for some things which are not really fitting into the framework of higher education. So, mind you that you are into an altogether different platform, wherein at present only 36% of our population, out of 100, only 36. That's also a bit far-fetched, I feel. So you are among those 36 fortunate people who are able to access to higher education in this country. And to have access to a facility like ours, I'm not telling we are having a state-of-the-art facility, no. But I have studied in college which is much more rural, and nothing wrong with rural, but much more less equipped, and I know the difference. So what I would like to say is that you're getting a great, great opportunity, and I suggest that you make use of it and demand more, so that your teachers who are willing to help you in becoming truly fitting to a higher education scenario will be eager to give you further. And they are resourceful, they are able to, Dr. Jimmy, Dr. Sibi, all of them are very, very resourceful people in your department. And it is only up to you to demand and get more and optimize the opportunities. I thank also our resource person, Dr. Deepak, for being available. Great that I think there will be many more occasions. And I congratulate the department for this initiative and tremendous work you're doing to make this such platforms available for the students. Greenhorns shows inexperience, but then I hope that gradually you will become thorough people with great experience, and that is awaiting you. So, bright future is awaiting you, a brave new world we are facing right now in spite of the great stress we are facing around us with the COVID. I wish you the very best. I wish the gathering and the process and the program the very best. May the Sacred Heart of Jesus bless us all. Thank you.
Thank you, Father, for that wonderful presidential address. Now it's time to hear our great speaker, Dr. Deepak Nayak, who is associate at Neuroscience, Indian Institute of Science. He did his master in physics from Indian Institute of Technology, Madras, and then went on to do his PhD at Leibniz Institute of Neurobiology. It's a biophysics field that he had done his PhD in neuroscience, spectroscopy, and microscopy. Then he went to do his postdoctoral research with Marie Curie Fellowship and CNRS Fellowship at University of Bordeaux. Deepak Nayak then joined Indian Institute of Science, and he has more than 40 quality publications in journals like Nature Biology and Proceedings of National Academy of Sciences. So these are really high quality journals where he's publishing with projects and 5 crores that he has handled and he's handling. He's also invited not only to institutions, but to industries like Carl Zeiss, where he goes and gives his or shares his knowledge. He has got many fellowships. So he has got a fellowship from University of McGill, Canada, then Ramalingaswamy Fellowship from DBT India, Department of Biotechnology India, and the very prestigious Innovative Young Biotechnologist Award from Department of Biotechnology India. And he's not only a very good researcher, but an extremely good teacher. And that's why the very prestigious Preeti Shankar Teaching Award in Indian Institute of Science was awarded to him 2 years back. So we are very fortunate to have a speaker like with us to give us or show us the light towards the field of research that he is doing. So the Greenhorn meeting is actually a meeting that we have organized for first year students, which will motivate them. After listening to these talks, you will know these kinds of researches also exist, and maybe in future you might take this as a career and move forward. So thank you very much. So he has a 11-page CV, and if I start to read that, the whole time will be over. So I have done a great injustice to Deepak by shortening it and telling it in a very short way. So Deepak, anyway, thank you very much for coming here, accepting our invitation, though you are in a very busy time schedule these days. Thank you very much and welcome, and let's hear from you.
Thank you, Jimmy. Before I start, I mean, I really would like to appreciate this initiative taken by Dr. Sebastian and the Sacred Hearts. And when Jimmy had called me to talk about this meeting, we just talked about the years back when we were like doing our degree and masters. And at that time, we would have done anything to attend meetings like this. Now the technology has improved. We have a lot of opportunities to attend these meetings, and I really urge you to go and see the things that novel things that are happening around the world, and also try to see, because our sometimes our environment limits us from trying to find new ways, but you hear from more people, you are able to find new ways to explore and new technologies and new avenues that you could explore in your future. So, and once more, before I start, I would really like to thank Principal Reverend Father Dr. Prashant head of the department for physics, Dr. C.B. Matthew, and most of all, Dr. Jimmy Sebastian, who has kindly given me the invitation. And it was just nothing. He didn't have to coerce me or anything. It was just my pleasure to be part of this. I'll try my best to, take you through some of the things that is very interesting in the field that we are working in. I'll try to keep it as simple as possible. But if there is anything that you feel like asking, please don't hesitate. Please do stop me, and you can feel free to ask. Okay, with that, I will start sharing my presentation. Let's see. So I hope this screen is visible to everyone, right? Hello?
No, Deepak, I think you are visible, but the screen is not visible.
Okay, so I will just try to share it once more.
Yes, that would be nice.
Sharing. Is it visible now? Hello?
Yes, yes.
Visible.
Okay, good. Okay, so I hope, may I start? Is it okay?
Yes, please. Yeah,
Okay.
go ahead.
So, my dear young future budding scientists and engineers and entrepreneurs, so this, I mean, I am very happy, and in fact, I am really proud to be part of this meeting and try to give my own few cents of what I am doing and what I am trying to accomplish in my lab. And but I will try to keep things very simple and as much as I can, and I will try to explain some of what does it mean by why are we doing that, and what is the implication of the research that is happening in this field. So basically, you will see that the title of the talk that I have here is High Resolution Microscopy and Nanoscale Biology. To understand this a bit better, I'll just try to take you to understand what does it mean by nanoscale. So if you are, so you know, when we are trying to think about the materials, we try to think about things that is visible to our eye. Now, the nanoscale is a special regime, where, like, if you are trying to reduce, either the length or breadth or the volume, in a size range of around 1 to 100 nanometers. This is very interesting because this is the scale. This is not a purely an atomic scale, and this is also not a scale where you have macroscopic structures. So this is something. This is a special scale that we physicists love to call it as a mesoscopic scale. Or so the rules in this special regime is actually defined by the mesoscopic physics. So what I mean by the mesoscopic observations are those observations that are sized between microscope, or like say atomic and molecular, to macroscopic, where you start seeing structures, with the help of eye and simple microscopes. And this is a very, very interdisciplinary field. I'll come to that. But before that, let's just try to understand, because since we are dealing with biology, let's try to understand, this simple slide that we have, what the size of living, what is the different dimensions that we have where the life actually manifests, So this is like, say, blue whales, which is like, a few tens of meters long on one side. You have human, who's approximately around 1 to 2 on an average, around 1.5 to 2 meters in their height. And, on the other hand, you have atoms, which are like, say, few angstrom, and, amino acids, which is dimensions of around 1 nanometer. Okay. So you will see that the biology actually means, you have to really understand atoms need to make molecules, molecules need to come together to make bigger molecules. They start to make functional complexes and simple life forms like viruses, which should now start working independently. And then these should again go up and start making more complex organisms like bacteria, and even more complex like plant and animal cells. And you will see that as the size scales increases, the complexity of these organisms actually increase quite a lot. And if you look at, the what, how we can see these things, we can, with our human eye, we can see around, like, say, precision of around half, few, about a fraction of a millimeter, like, 2 to 3 millimeters. And below that, we need different kinds of, systems to see. In some cases, we need light microscopes. And when we go to, like, say, around the scale of molecules, we need an electron microscope, which is even quite advanced to see this and study these structures. The question is that why do we want to study these structures? Okay. So, like I told you, so when we try to think about a cell or a living system, we have to actually think about what are the structural or small units that makes up a living system. Like I told you, if you have a kind of a cell, which is a few micrometers in their size, you have even substructures which actually are the functional components, like, that you make it up. So you have, these are some of the common nanoscale biomolecules that will come into the picture. You have, like, say, DNA, you have the antibodies, these are the terms that you hear. You have chemicals like glucose, you have proteins, you have viruses. So these can come together, for example, lipid molecules or fat molecules can come together and start making structures like liposomes, or you can have, they can be organized differently to make, structures known as dendrimers, and all of these are, like, say, in the scales of around biomolecules. So on one side, you have a very high organization which is created by nature, and second case, we can alternatively try to mimic nature using synthetic and chemical biology to create structures which is very much organized and start behaving like some very to the nanoscale biomolecules. But however, this could be synthesized in the lab. So that's where the center, the culmination of the nanobiology lies, because you actually try to observe of the biological systems at its finest resolution. That means at the level of molecules, and how these molecules come together to create responses at different scales. It could be a response at the cellular level, or it could be action of lot of cells, creating at the much more at a larger level, like, say, contraction of muscles or movement of muscles allowing you to walk. So these both happens, start with the changes in molecules, but the changes, the observations are happening at completely different dimensions. Okay. So, and how can we study this? We can study this using, to study this, we need a lot of things in hand. You should have systems to observe this system. So that means you should be able to observe the biology at the level of few tens of nanometers. Then you should have an ability to understand how the synthesis, organization, and fabrication of the materials happen, both at the biological level and also at the artificial level, because studying this at the biological level would enable us to address this at the level of artificial, so that we can manipulate, we can try to create these things outside with the in the lab, by trying to mimic the nature. And next thing is that we need to, because all the things, in a biology, one of the biggest things are the degrees of freedom. That means you have a lot of variability in how these cells happen. Like say, one thing I could like to tell you is that, I hope everybody at one point has heard about Alzheimer's disease, which is a major neurodegenerative disease that's happening. So some of the familial variants, so genetic variants of this disease can happen by change in one or two amino acid differences in the protein, amyloid precursor protein. So this is a protein which has around hundreds of amino acids in it, and and this is made from a genetic sequence which has several nucleotides. And if you have one point mutation, one amino acid or two amino acids are changed, this will start having a behavior which is different from its regular natural behavior, and this will start having the disease or progress. It will progress towards us having the disease. This is one example. So that means that, so this is not just looking at nano and biology, but it is culmination of lot of things. You have, you need chemistry to really understand the chemical reactions and modify the molecules. You need physics to understand and to measure the changes that is happening. You need chemical engineering to really understand and alter the behavior of molecules at fine scales. You need nanotechnology. That means you need to be able to probe this biological systems at few nanometer level and be able to observe them. And you need to engineer things, both you need to engineer biology to presume or to do a specific function. You need to characterize the biochemistry behind this, and you need to have applied mathematics and computation to really predict how a model would behave. And all this would in a way explain biology both at the nanoscale. And it is not only that we need to explain this at the nanoscale, but we also try to see how this would build up so that we can explain larger systems like action, diseases, behavior of a human being, etc. I think I could start by giving a very, very simple example. Okay, it's about how brain process information. Because coming from Center for Neuroscience, it is much easier for me to start with something which I am little bit familiar with. So now take the case where somebody rings a doorbell. So what really happens here? So here it's again very straightforward. The sound is transmitted as sound waves from the environment, and these are gathered by the outer ear. So this information easily you can find from, if you type how hearing happens, this information would come up. So basically what happens here is that the sound waves are gathered by our outer ear and sent to ear canal or the eardrum. So now this sound waves, whatever it is, like the doorbell, what we heard, it causes the vibrations in our eardrum, and which allows, a few bones in our middle ear to move. And now when the movement of this bone happens, so there is some fluid which is in the inner ear, which is preserved there, and that it affects the motion of this fluid. And now once this moves, this starts affecting the inner hair cells which are in our inner ear. Okay, and now this movement will be converted into electrical signals, and these electrical signals are transmitted into the hearing and up from nerve to the brain, where they are interpreted as sound, which is a doorbell. And like now, if you are lazy, you just sit at the couch. If you are really want to know who it is and why this bell was rung, your body will start moving out of the couch or from wherever you are towards the door to open and set you for the next step of interaction. You would think, what is funny about this? I mean, this is something that we do every day, right? But now we need to come back at different scales of how this would happen. So when the sound is converted to electrical energy, the molecules, these are the molecules which are called as the channels. They actually send positively charged ions from outside of your cell to inside, making cell more positive. So this would allow propagation of potentials, and they start activating the neuronal small neuronal compartments like synaptic, presynaptic compartment, or like say or the cell body. Now these signals will come to the neuron, and at the cell body, or at the place you have the soma or cell body, it will actually think, okay, should this signal be converted? Is this the right signal? Should I transmit this signal to next neuron? So then if it is the right signal, it will transmit to the next neuron, creating a network of neurons called as the local circuits. Now these local circuits start giving the information to your brain, which is like say around few tens of centimeters in diameter, which will allow you to take the final decision. And you have to remember, from the time that you heard the doorbell till your decision that you are supposed to take a decision whether to move or not, this happens probably in few millisecond time scales, which is across a dimension that is from 1 nanometer to like say few centimeters. So if I want to compare this in real, what is the change in this dimension? Okay, so it is the differences that we have for the size of the earth is the size of a tennis ball. You can assume the tennis ball to be here and the size of the earth to be here. So all the signals that we have has to be computed, passed on to take a right decision in a small time to give you to process this information. Now, how can we do this? How is it possible to observe these kind of changes that would happen at channels? Because the channels are the tiniest level where we start this, right? So we can do that using microscopy. So we can see by naked eye, I told you we can see a fraction of millimeters. And you have to remember that it's around 1600, that's around 400 500 years back, they actually identified the first microscope, which had a magnification of around 9 times. Then I think many of you have heard about Leeuwenhoek, who had actually improved the microscope. He actually reached a resolution of around 200 times. Okay, where he tried to understand the microbes that is in the teeth, which is in the mouth. So he actually found that there are different kinds of shapes for these microbes. And what was also interesting for him is that he also found that he can actually see molecules, or the microbes actually moving in the cell randomly. Okay. Now, then the compound microscopes had come up, which actually improved the magnification and resolution 1,000 times as that of the naked eye. And then those people who have done physics and engineering would know that Ernst Abbe actually said that you have a limit of diffraction, physical diffraction, below which we cannot resolve things. And surprisingly, if you see here, that is actually coinciding very close with the size of the cell. I think it is very clearly, I can bring back here. So it's around the size of like say around 1 micron. So if you are trying to, this is an example. On the right side, what you have is an example of a cellular structure, or a cellular structure called a cytoskeletal protein. It's like the skeleton of a cell. And this is actually a combination of, this is a polymer of monomers. Okay. If you are trying to increase the magnification, you don't really get much information, but you only start getting blurrier and blurrier pictures. So what have people done to improve this? So they try to understand, last century they tried to improve a lot in optics, they tried to improve a lot in engineering, they try to improve the quality of the probes, they try to improve, whether you can try to illuminate differently. So all of these are many, many different methods, all try to improve the resolution little by little to see whether it is possible to see at the nanoscale, but just using the light microscopy. The thing about using light microscopy is very interesting, because if you want to really use, if you want to really see at the best possible resolution, you can use shorter and shorter wavelengths, right? But the problem is that as shorter you go, this is not compatible with light. For example, electron microscopy cannot be used to observe living things. So, but light is compatible, so it is much more easier to observe systems using light, because it keeps the biological systems or cells alive. So that's why this unending, effort to increase the resolution that we use with the light microscopy methods. So then a body of work from the group of Eric Betzig, Stefan Hell, and William Mornier actually created a new branch of science called as a super resolution microscopy. So we are familiar with with the word super. I mean, I will try to use this in a very different context. I think there is nobody in the audience who might not have heard about superheroes. So it could be a Superman, it could be Batman, or it could even be a Hanuman. But the beauty is that all these guys can take, they have a capability, and they can take an amount which can bypass or surpass the way a regular human being can do. And many of times they have this unending ability to supersede, unending ability to extend their powers. For example, you have Superman can do things, Hanuman take a mountain. These are things that normal things can do. So similarly, super resolution microscopy is a branch of microscopy where we can try to understand the structures at the level of molecules, but by preserving the state of the living state of whatever system that we are looking at. Now, let's see. So now I will try to introduce you to a very small part of the work that is happening in our synapse using the super resolution microscopy to give you a small idea, small feel about how this is. For example, this is a neuron. These are the neurons that you have, nerve cells that you have in your brain. So you have this is the soma or the cell body, and you have this nice dendritic structures. And if you look very closely, this is the dendritic structure, and you have this mushrooms type structures that you see here, right? These are the places where the synapses is there. So our brain contains around 100 million neurons, but each neuron can make up to 1,000 to 10,000 synapses, which means that our brain has around close to 100 billion synapses, which is quite a huge number. But now if you are trying to look at the real structure of the synapses, that's what you see here by the electron microscopy. I have marked it with red. Okay, so you have this is the postsynaptic, or this mushroom type structure that you see here is this structure, and what you see here is a small connection which is in this black. Okay, and this is around 200 nanometers. Now I have to tell you that now every time, even right now when I am talking to you, the synapses in your brain are being altered. So they are actually changing, where the number of molecules, the density of proteins, the interaction between these compartments and the compartments around it is in a constant change. And let me tell you, this is actually modulated or mediated by billions of these tiny structures, which are around 200 nanometers in their dimension, in their size. Now, what I have done is that what I'm trying to do here is that I have tried to pick one protein molecule where we know that can change the ability with which synapse can capture signals. Okay. For example, you have a molecule that can allow charges from the outside of the cell to come into inside of the cell. These are called receptors or neurotransmitter receptors. So in this particular kind of synapses, if you have large number of receptors or if you have less number of receptors, will tell you what is the signaling state of the synapses. But it's not only that, it's also about how these molecules are clustered, because if these molecules are loosely packed, you will not have a directionality in the signaling. So our lab went on to understand, how we can study this organization at the nanoscale. And this is what we see. So this is a brain, this is a neuronal cell which is in a place called as hippocampus, but this model organism is mouse or rodent. And these structures that you see here, these are synaptic structures. These are the places where synapses are. But the scale, the resolution that we have here is for that of a regular microscope, whose resolution is around 200 to 300 nanometers. So now what we try to do is that we try to observe this in live, and we try to induce a kind of physical phenomenon called as photoactivation, I would say. But what is interesting now is that you can start seeing molecules. You can start seeing this green light which is running on the red, right? Which is the neuron. Just one second, my charge is getting finished. I'll just plug the. So I hope you can hear me, right? Someone give me a yes, please.
Yes.
Yes.
Okay, okay. So you have this large number of molecules which are running on this neuronal dendritic shaft. So now we try to see what can we do with these signals. So we try to use large scale computation methods to really predict what would be the center of emission of this fluorescence, okay, or the emission that is happening. So, and we recreate an image like this. Now, if you look very closely at some of the sections, you can see that the image that you have below is very wide, vastly improved. And you look at this, you can see the color means that white color means you have high density of molecules, and black color means you have less density of molecules. You can see that these molecules are clustered into these small regions, which is marked by white to red regions. And each of the structures are around 50 to 100 nanometers in diameter. So we have reached nanoscale here. Okay. Now the beauty here that you need to remember is that when I am talking to you, the molecules are moving in your brain like this. Okay. Sometimes they come, they kind of get stuck in this kind of thing. So to understand that, we try to model each of this movement of these molecules in the cell as a random walk phenomenon, like how these molecules would move step by step. And this is what we see. So this is, I am just trying to zoom into one example. You can see that these molecules just moving quickly, and they tend to get, trapped, they tend to get clustered, and then they move out and move away. So what happens is that we understand that, the behavior of these molecules, which is actually coding the way our memory, the way we interface, with the world, and computation is similarly like us, because if you try to track our movement in a daytime, we are at home. So if you track our movement, we will see that we are actually stuck at home. Then we will take a bus or a car or a scooter and come to the college, where we are stuck at the college till at 4-5 o'clock in the evening. Then we move back by bus and then back at home again. So we will see that, the steps, the ability or the time that we stay at each places is defined by the amount of steps or amount of time that we interact with that environment. The beauty is that similar to that, we do that for each person, we can do that for each molecules inside a neuron. Okay. Now, what all this tells is that, like we happen like we do in real life, molecules also move, stay, and function, and all about is to be at the right place at the right time. So to understand this is one of the fundamental questions that we have in neuroscience, which will enable us to understand, not only the way we code memories and retrieve memories, but also about how many neurodegenerative diseases like Alzheimer's, Parkinson's, dementia progress. So what did we, I think with these things, I will try to slowly wrap up. So what did our lab trying to study, and what did we contribute to the knowledge that we had? So I would like to start like this. When I was in school, people actually thought that synapse was a very stable structure, which is stable for like days and, hours and days long. When I was in college, okay, people actually found that there could be inserts, that molecules could come in and go from this at the level, or in the matter of like say minutes. In 2002, there was this idea came up that molecules can not only move, but they can through the pathways that they have in the cell, but they can also move on the membrane, because every membrane is like a fluid. So it allows the diffusion of molecules on the membrane. So now this allowing of movement on the membrane will allow this to be exchanged at the matter of minutes to seconds. Okay. Now from 2002 to 2013, the model existed that okay, there can be exchanges in the matter of seconds to minutes that would actually control the organization of molecules at the synapses. And what we showed using our techniques is that there can be clusters of these molecules which are at nanoscale, and this exchange between molecules inside the cluster and outside the cluster can happen at the scale of milliseconds, the time scale at which the memory is coded or memory is retrieved. Okay, this is a very important information, and now the model thus have been altered accordingly to say that all these processes coexist, and our work is trying to understand the small regime in how the organization and transport at the millisecond time scale and nanosecond spatial scale contribute to the proper functioning of the brain and thus the proper functioning of organism. Now, before I end, I would like to say that this is only one of the example. You can actually use this to make, this combination of biology and nanoscale can be used for several things. Understanding this would also, it can enable better computation, better sensors. You can try to have better models of about creating and using energy. You can try to have models, even models that we could use in the finance regime or mathematics to see how we should dissipate, how we should spend energy, currencies, and how we should spend market. A lot of information can be understood just by observing these molecules and their behavior at nanoscale. Last but not least, I would really like to thank my group and also all the funding agencies that fund us, including Government of India and other funding agencies. So without these guys, I cannot do any of the things that I am presenting. And they have been a tremendous support and a constant force for me and others to move forward in the project that we are doing. Last but not least, thanks a lot for your attention, and I will stop with the picture of IAC. And this is the main building of IAC. It is a very picturesque campus. And every time I come and stay and visit this small place, I always think that there are almost majority of the scientific decisions that we have in India is actually influenced quite a lot by the decisions and the people that have worked in IAC, which is very it makes a very proud to be here. And I hope that each and every one of you can grow up and find your way to move forward. Thanks a lot. So Jimmy,
thank you, Deepak. Thank you, Deepak. It was an extremely wonderful session. I mean, as you know, there's no doubt that you are a wonderful teacher. I mean, I was myself motivated listening to it. So I'm sure the students have benefited a lot out of it. I would like to tell the students about your career journey. As I know.
Yes, yes, I mean,
college, and then you have reached this level of reputation.
Yes. Thanks.
Maybe
thanks.
Tell that will actually help the students to also target.
Sure. So, like, I think I yeah, like when Dr. Sebastian, when Jimmy started, he told you, like, we studied together, and I have to tell you that both of us do come from, not very big towns in India. I come from a small place in Alapi called as, a small village called as in Mararikulam, which is in a coastal part of Kerala, and I started my schooling in a very vernacular school. I started in a regular government school that we had in Mararikulam, and it was a regular school. It had up to fourth standard, fifth standard, and after that I moved to a school in a little bit bigger place, and then I moved to a place called a Sainik school. Okay, which allowed me to improve myself in both in confidence and also about how I present and how I interact with people. But most important was not that. So when I came back, when I finished my Sainik school, I was extremely motivated to join defense and army and so on. And it did not work out the way I want. And I was trying to start thinking because I didn't have a backup plan at that point. And I thought, okay, man, I should start thinking how I should do about it. And I came across, there was a small a set of books which my mom has actually bought it from someplace. It was a book called as Basic Radio. It's a very old book, and I don't know whether it is published anymore here. So it actually talks about about how the basic principles of electricity and basic principles of about animal electricity and so on, which actually made me fascinated towards, okay, I thought it was a time my after the 12th standard or pre-degree time has finished, and thought that okay, this might be a good option, would be either to take a physics or something in applied physics to start with. Okay, and that is when I start getting really interested in physics. So many people would think that okay, if you don't have a plan in 8th standard or 9th standard, or 10th standard or 11th standard, we should be worried. I mean, you don't have to be worried about anything. It is because it is your interest that has to guide you, whether it is you becoming an academician or whether you are becoming a social servant. You have to relay, you have to trust your instincts here and take that course. Take the course of that action, because that will keep you motivated throughout your life. So now, after reaching, after this, I joined a local college, a college which is SN College Chertala, or which is again not very far from my village, and I finished my degree over there. And then I had a chance to, I thought, okay, now I should try to pursue something higher. Okay, and I tried. Then again, the question had come: should I come out and try to do something like an applied things like MCA, like which deals with computation, or should I go for hardware engineering, or should I try something different? I thought, no, I will still stick to fundamental science, because I started slowly getting a kick of optics and how optical systems behave and so on. So I thought maybe it's a good idea to try different places. And like that, I had a chance to write the IIT exam and had the luck to get through. And when I reached IIT, I found that almost everybody was better than me, and it was really a surprising thing, and there was high level of competition. And I could see that everybody comes with a kind of intellect that was very stimulating. And it was like every person has had something in them, and a spark for a different subject. Some people were interested in astrophysics, some people were interested in theoretical physics, some people were interested, people like me, I was interested in optics and little bit of biophysics from that time. But the fact was that everybody whom I interacted with was able to bring their little perspectives to the same question that we were looking at. And so that made me think even go further, because I was thinking that okay, I should have a way of my thought process is actually has to be okay, I understand something, then that's a rigid thought. And I thought it's a good time, good interaction that we had. I started keeping myself more open, because the more time I interacted, I started learning a little bit more than that I knew from previous day or previous discussion that we had. So that actually gave me much more motivation, and wanted to see whether I could do something bit more nicely, something I could get an information, something nice on my particular interest, which was a combination of optics and something with something to on applied biology. So I tried to move on and tried to start in a lab, spectroscopy lab, which was trying to understand, the time scales at 10 raised to minus 18 seconds. But the problem was that I was very much interested in the work, but the time for planning and executing the experiment was really huge. And I thought that maybe I should move on to something more challenging. And I had a chance to work with at that time my colleague. She was actually starting to work in neuroscience field. And then when I tried to check that, I got a position. They actually invited me to join their lab. That is where I did my PhD. It's a lab in Germany, and they gave us, like every PhD students in Germany get, we also get the scholarship to pursue this, and then try to combine, the higher order spectroscopy to combine higher order spectroscopy to understand the biophysics. And that's where we started tried getting more and more interested in neuroscience and more interested in the synapses. And then we moved on to after the PhD, where I moved on to a lab which was in a French town called as Bordeaux, which is actually very famous for wines, and also very famous for single molecule fluorescence. It was one of the places where they actually recorded the first single molecule fluorescence. And also it was the place where they actually found that the molecules can diffuse on the membrane. So my job was to really combine these two fields, because I knew that this has a particular progress. I mean, this has a particular implication to understand the biology at its best scales. So then I tried to work on this, tried to combine. So at the time when I started my postdoc, they had actually developed the super resolution imaging, and me along with my French colleagues started working on it. And we were one of the first groups to record single molecule fluorescence and single molecule imaging from live, at the super resolution scale in live neurons. And some of the things that we have developed over that time had actually now became the state of the art, and we are very proud of what we have done over there. And as I was moving forward, I tried to come and, after the first part of the thing, establish the things in France. Slowly I wanted to thought, okay, it's a good time to try and move back to our country, because I really wanted to try what I can contribute. And if I thought, okay, if I am a failure, I still have a chance to go back, learn something new and come back. But if I can find a little bit of success, at least I can train a few set of people who can go and be better than me in future. And thus I am here, and I joined IAC as a faculty, and where I am still pursuing, trying to understand this particular detail. I hope it was not too long, but I hope it's okay. Thanks for hearing it.
No, thank you very much, Dr. Deepak. The person with whom he was inspired is another great researcher. She's Mini, Dr. Mini, who is also pursuing this field of research. And later on, they married, and they have 3 wonderful kids. So both Deepak and Mini are doing extremely good in research. And thank you very much, Deepak, for this wonderful talk. And now let's see if the students have any doubts, so we can ask them. So, yeah, any questions? Can you please either write it down in the chat box, or you can ask yourself. It's a wonderful opportunity. You get to Indian Institute of Science.
I think I was too much, or I made them complicated, or
No. I think the thing is with the students in mostly from India, or I would say South India, is that they are very afraid to ask questions. I mean, they are very bright. Many of them are very bright, but some of them are, like one of the questions is about tachyons. Arundhati had asked about tachyons. Tachyons is, I think, out of the topic of today's talk.
But I think it's a great thing, and I think Jimmy can also help. I think if you talk about tachyons, these are the molecules that supposed to move faster than light. Are we talking about the same tachyon?
Yes.
It's not. I mean, one thing is that we still do not know the experimental evidences. I am still not sure about whether there is a clear experimental evidence of their existence. But theoretically, there are predictions that could predict that could show that they could exist. But unless anything is really found, it still remains an open question, right? Because there are so many the things at the atomic and subatomic scale which is not yet clear to us. So tachyons is still, for me, I think for many of us, is an open question.
I mean, that's a biophysicist really speaking about tachyons, which is in the quantum field theory. So Deepak, I mean, that shows his intelligence and his wide knowledge.
No, I think I actually feel that Jimmy would have been the best person to answer this, but
no. So very nice, Deepak. I mean, very good. So there is another question from Professor Ante Joseph. Now his question is, sir, can you mention something about the fluorescence proteins that you are using?
Oh yes, this is very interesting, because you have to understand, in the last 25 years, different aspects of nanotechnology has contributed to several Nobel Prizes, and one of the Nobel Prizes is actually was given for the development of fluorescent proteins. So I think that I also see another question from another person also in the same way. So basically, there are a lot of fluorescent proteins which are autofluorescent, which can fluoresce and or chemiluminescent. So basically, what happens is that they either can absorb light and emit light as fluorescence, or they can absorb, they can take chemical, they can sense chemical changes and emit light. And I think one of the famous things is actually from the jellyfish, and you could, this is called as Aquaria Victoria. So this is a jellyfish that you will see in the sea. And the first fluorescent protein was actually extracted from the jellyfish. So by a Japanese scientist. So then the people have actually tried to understand this protein. The beauty of this protein was that because it has a genetic code, you could directly express it in an organism. Okay, so now the people have tried to identify several fluorescent proteins from coral reefs, for example, from different kind of algae, from the pollen grains and from the plant zone, and all of it actually contributes a large family of molecules called as the fluorescent proteins or fluorescent pigments. Okay, and this is very important, and in our case we actually use them to tag molecules of interest. And even in the fluorescent proteins are also like different classes. Some fluorescent molecules can absorb, some fluorescent proteins can absorb light and emit lights at a longer wavelength, while some molecules can also change their emission and absorption spectra upon the absorption of blue light. These lights are called as photoconvertible. These molecules are called as photoactivable or photoconvertible fluorescent proteins. And this is also widely used for different kind of purposes in trying to observe the living system, both the regular fluorescent proteins and the photoconvertible fluorescent proteins. I hope I answer your question.
Thank you, thank you, Dr. Deepak. There is another very interesting question from K. Manushri. Sir, I saw the movie Life of Pi. At night, the sea animals will glow with multiple colors.
Yes.
Is possibilities or reality that they are existing?
See, that's exactly what I have been trying to tell you, because I saw the flash of that question while I was talking. Yes, I think many of the sea animals have this ability to sense, be either photoluminescent or chemiluminescent. That's what I have been trying to tell you. The first fluorescent proteins that we originally extracted was from jellyfish, which is a sea animal. So yes, in a way, this sea animal glowing is a reality. It is not a Hollywood trick or a Bollywood trick. So you can believe that.
Very good, very nice.
Yeah.
Now, any more questions? May I know who is speaking?
Yeah, thank you, Manushri.
Thank you very much, sir. Welcome.
So, Jimmy, I can't hear you.
Yes, there is one more question.
Yes.
Neha George, she has written, sir, which is the better option to pursue among MSc and integrated PhD after BSc?
So it depends upon what you want. This is a question that, so the good thing about the integrated PhD in IAC is that there is a provision. You can actually, at the level of masters, you can actually try to think whether you should continue in your integrated PhD program or stop with masters and try to choose a different option. So I would rather say that integrated PhD post in that way is a very attractive program that gives you the flexibility to do the training in IASC. And the great thing about is that you have a choice at the level of masters what you should do with your future. So I would rather, at a personal level, I would rather go for integrated PhD program because of the offer it gives after the BSc. You can start early, and you can also try to take a decision 2 years in time whether you should continue for PhD or in a particular program at IASC, or you can try different options. While if you start PhD later, you have to complete masters and you have to start the PhD. In that case, you start and you can still choose, but it may have this additional training time that is required. If you're in an integrated PhD program, it's highly likely that you can finish in around 4 to 6 years. Okay. While if it is a full PhD program, that alone will take around 5 to 7 years on an average at IAC. So it's up to you to take a call on what kind of time frames, what kind of subject, and what kind of motivation you have to pursue these different fields.
Yes, very nice. Now there is another interesting question, a very important one. I mean, from Merlin Baby. She's asking, sir, can we expect that we could stop diseases such as Alzheimer's? I mean, Dr. Deepak is working in the field of Alzheimer's. Yeah, that's a very, very, very difficult and a very important field. So I think the right person.
Yeah, it's a very, very interesting and important question. Okay, so I will just try to give you some little facts before I start. So if you look at Alzheimer's, it is one of the, if there is any disease that has been funded most in the world with a particular cause other than the cancer, it should be Alzheimer's. So it is the people are funding a lot on Alzheimer's research. But the problem or the issue that we see is that how we are trying to understand the diseases, or what are the problems that we see when the disease progress. So if you try to think of a disease, this disease could happen either as a genetic cause, or it could also happen as a result of how we interact with our environment. For example, if you have cardiovascular diseases or vasculature diseases, you have high chance of getting Alzheimer's. So that means that it can never be considered as a disorder which has one single cause. Okay, which means there is a lot of things we have to look at, when we try to understand the disease at its fundamental level. But despite that, there have been some, particular breakthroughs, to slow down the Alzheimer's, to kind of, rescue some of the functional deficits that we would have at the Alzheimer's. But I think eventually, I feel that the field that we are currently working on, the idea to understand the biology, the nanoscale biology, where we can understand the chemical reactions at the nanoscale, and trying to regulate these chemical reactions holds a very, very big potential in trying to regulate diseases like Alzheimer's and so on. It's a very new field, so that we can start devising smart medicine. It's a field, I would call it as a smart medicine. Rather than treating the disease, you actually start trying to manipulate what could cause the disease. That way, we never reach the state where the disease would happen. And nanoscale, if you try to, the nanobiology, if you try to look at it, has that potential. So if you look at the Alzheimer treatment and the strategy right now, yeah, there has been several progress actually being made. But if you sincerely ask me if any cure has been found yet, no, not much, because after 2003, no any additional drugs that has been identified that could actually cure or that could actually slow down Alzheimer's, except the one that was approved in Chinese market last year. So, which is not approved by American FDA. It's the American Medical Foundation kind of, the validation. So, if you ask, yes, there is a lot of potential, but still there is a lot to know, because you see, you have to understand the reactions, the proteins and the molecules that is in our body stay for, like, say, hours to probably days. The disease of Alzheimer's actually occur as a matter of accumulation of the toxic pathways or toxic products over years and years in our lifetime. It occurs towards the old age. So the idea is to really understand the fundamental biology behind it. And I think once we start understanding it a bit better, there are ways to address it in much more much more precise manner, or much more tailored manner. So my answer is that right now, yeah, we have drugs which would aid you in a recovery or like say partial recovery and all, but we do not have anything that would cure at the moment. But yeah, the future is green, future is bright. So maybe, Merlin, you could do it too. you could find a cure too.
Very good. Very nice. I think Merlin is highly motivated after listening this. Now there is another question. I think there are a lot of questions. One is about whether in animals or all organisms the stimulus processing is the same or different. I mean, that is one question from Arundhati. And the other one is actually a very difficult one to explain, I think, in a very short talk. And that's about elaborate conceptual side of technique super resolution microscopy. So I think you need some slides and, to really get the idea through. But I mean, I put it forward to Deepak. I mean, he's very bright. So he will find a way out of it to explain it.
Yeah, okay. I will try to do my little thing. So the first question was that, whether the sensory things in organisms are same or different. Okay. So basically, you have to realize that as the organisms evolved, we also evolved our organ systems and the cells that we have in our body. So, for example, if you try to take the nervous system of a worm, it has only around 306 or 300 less than 400 neurons. Understood? So that actually constitutes the complete nervous system of a worm. But if you are trying to take humans, it's around millions of neurons which has to interact. That would also mean that there is an evolutionary complexity associated with it. So which means that as the organisms evolve, you have to interface with multiple, environment, to really survive. For worm, it can survive by finding nutrients, trying to reproduce, and there are the parameters that it has to take to live is much less than that we have. So as an individual like us, we have more emotions like fear. We have emotions like we have cognitive thought processes like learning and memory. We have sensory processes that has to be integrated, and which should mean that the system is actually improved and evolved for better efficiency and better specificity. So an example of this would be is that if you look at very, very lower order organisms, they have a lot of electrical synapses. That means the complete connectivity is through electrical, it's completely electrical, the synapses. And as the systems evolve, you have a system where it's a combination of electrical and chemical synapses. That means you have synapses that could work, both electrically and chemically and more. And that means you have, it's not complete electrical signals. You have an electrical signal, then a small chemical signal, which is again converted to electrical signals. The beauty here is that it will create a specificity. That means if you think electrical signals, it can flow forward and backward, right? The chemical signals cannot flow easily backward and forward. So one will be a donor reactant and other will be an acceptor. So which is very important. So that means you have a unidirectional signaling. And as the organisms evolve, like as we become human beings, we have more chemical synapses than electrical synapses. Okay, so if you try to look at is it working? Is the stimulus working the same way? It is. If you look at how we interact with external environment from different organisms, it may at once look very similar. But if you look at the molecular mechanisms and the mechanisms at the micron and nanoscale, it could be vastly different. Understood? So every system, every organism has its own way that it has adapted to live in the nature. From like say unicellular organisms to multicellular organisms like us. Does it answer the question? Yes.
Great.
Okay.
Great.
Yeah. Thanks, Arundhati. So now I think the next question that they asked was about the idea of about how we try to use super resolution microscopy. Okay. Just let me try to let me try to see if I can pull out one slide to help this issue. Give me 1 second. Okay. Yes. So let me. Okay. I will try to share application. Okay, so can you see this? What microscope does? a slide?
Yes.
Yes, so I think I will try to explain using a couple of slides. I hope it is not too much time. So anyway, so when the light passes through a lens or thing, it focus, and you have, so the what when it focus, because of the diffraction of the light, you are not going to get a point. The light is going to spread in all possible directions, and you will have a elliptical structure, like a rugby ball. That will be how the focus would be. Now the question is that how can we improve this thing? That actually tells you how can we improve the resolution. So one of the techniques that people try to use is what really happens when we do microscopy is that, we have a point source. Now it is coming through the objective, and it actually falls on a camera or a surface, and it creates patterns like this. These are called the airy rings. Now, if I am trying to put a line and try to measure the intensity, you will see that, this central ring this central ring that we have here holds around 84% of the intensity. Okay. So basically, a single point spread function looks like this. Okay. Now, what happens is that this is when a lot of molecules come together. Okay, so this is the resolution limit. This is like, say, here center of each molecules can be well resolved. Here what happens is that the molecules come closer and closer, and it is very hard to resolve these molecules, and there is a limit where you can resolve this, and that becomes the resolution limit. But when we try to look any of this ensemble of these molecules under a microscope, what actually happens is that lots and lots of molecules overlap, and we cannot resolve these molecules anymore, creating a big blur. So there are multiple ways where we can actually do that. So we can try to improve try to make smaller POSFs. Okay, we can try to improve selectively excite only a small plane of illumination, but none of this actually gives us unlimited resolution. But what could be done is to start playing with the sample that we are looking at. For example, you can try to break this diffraction barrier either using ensemble method or using single molecule method. So in the ensemble method, what happens is that, this is the fluorescent molecule or the fluorescence that we could have from a regular objective that is happening, and this is diffraction limited. Now what I try to do is that I only want to resolve a smaller structure. So what I can try to do is that send the molecules or send the fluorescence outside this black circle into a dark state. Okay, so how can we do that? So I will skip all these things, but I'll just come into the basics. Okay, so how I can do this is, so I have this green spot. What I'll try to do is that I will try to have another laser which is like a donut, which is like in Malayalam, in Kerala, you have seen the ulunu vada, right? Or donut. So it looks a pattern like this. Now I superpose this laser and this laser together. So this red laser will start quenching the fluorescence that we have in the green region, which reduces the size of point spread function. Now the point spread function becomes smaller, and we can start resolving things better. The beauty here is, this is a phenomenon, so this is what happens. This is the point spread function that we had. This is the donut function, and the more and more we increase, we can reduce the size of the central PSF function as much as we can, trying to make it as smaller as possible. So this is called a saturated depletion, and this is also a kind of similar process is also used to create lacing in laser systems. Okay. And the beauty about this is that the resolution of this depends on the saturation or the amount of saturation we can do or quenching that we can do on the central function. So we can say that there is an unlimited limit, and how much where we can go, this is unlimited. But still you need to remember everything has a limit, and the ability with which we can locate a molecule is limited by the Heisenberg's principles. Okay, that you need to remember, which is much order of magnitude lower than this. But this method right now has never been shown to go anywhere less than 1 nanometers, but that is too much. That is a good resolution to look at the behavior of the nanoscale. Now we can do the same thing using single molecule. So what we do here, I will just skip it. I will skip all of this, but make it very simple. So you have an ensemble, something a structure like this. Rather than observing all the molecules together, you start observing molecule one by one. Okay, you start just look at one molecule and try to observe it. And what we can try to do is that if this is the fluorescence emission of one molecule, you try to fit the center of this molecule. So you calculate with what error you can understand from the center from this fluorescence is originating. So for example, this gray curve is the like say the emission of the fluorescence from a diffraction limited object, and this orange curve is the error in calculating the center. So once we calculate this error, we can represent, rather than this fluorescent spot, we can represent it as this error, and this is called the localization precision. Okay, now we can repeat this process. Okay, I will just see. So you have this thing, you have some molecules, you just locate the center, you start creating a better image. Okay, now this is what you see. So you try to repeat this, you start getting better structures which was not resolved in regular light microscopy images. Okay, and I can also give you one example here. So one regular example. So this is the cytoskeleton of the cell. Now I start inducing the chemistry with which it can go into single molecules like stars. Then you try to fit the center of each of the stars and create a structure, and your resolution, voila, has improved 20 times. So you can start seeing this. For example, if you try to look at this part, okay, or like this part, you can start seeing very well resolved structures, which where multiple lines starts overlapping, which is not very visible, let's say at this point. Okay, this is a second way that we can do super resolution. There are also other ways we can do super resolution, but at least this will help you to form an idea on how you should try to search in Google and move forward. Okay, I hope it helps you understand the question. Thanks.
Thank you, Deepak. That was wonderful. I didn't think that you will put out the slides. The teacher in him and the interest for optics, that actually, he doesn't want to leave that question. Very nice, very nice. Then there is one more question. This is about from Komal Vittal. Sir, can you please tell me how can I become ISRO scientist?
I think this is a very interesting and very complex question, because I have never tried to try to become ISRO thing, but couple of our friends are ISRO scientists. So I think there are different ways in which you can become an ISRO scientist. Okay. One thing you have to realize is that the ISRO is recruiting engineers with different background to become like applied scientists. They are also recruiting fundamental scientists from biology, chemistry, and physics. They're also recruiting people who have even done classical things like biology, and to some extent I should also say economics, because these people are very good mathematicians. Okay, so the ISRO has the opportunity. So I think most of the ISRO positions, as I know, are more focused towards non-biological thing. It does not mean they are not in. There is a small fraction who are working on it, but a large part of the ISRO research is towards the development of novel technologies, which would enable you to do next generation space research. Understand? So exobiology, or which is considered as a biology which is outside Earth, is not of a very big interest as of now in India, though NASA and the other space agencies has a big unit for exobiology. But what is important here is that whatever field you choose, you have a potential to go to and try to do space research in India, because they actually specifically ask for people to join their team with different skills. One of my students who was from chemistry background is currently in ISRO. He has done his master's in chemistry. One of our own the institute mates who has done masters in science with us, he has actually tried to become a leading scientist in ISRO in Trivandrum. There is also Indian Institute of Space Technology, which also allows you to do space research, and which involves a lot of mathematics, computation, and design technologies. So there are people who have done PhD in IITs who have gone on to join this IAST. So it's not about what course you should do. So whatever codes, what we are doing, we have to see which of, whether all, which would, how many of them would fit in the larger purview of ISRO. That would enable you to choose the position. But I am pretty sure that if you are able to contact one of these people, and most of the scientists who are there will be glad to help you out, and they will be able to write mail and just give you some better suggestions than I have given. But the thing is, no, it's not about choosing biology, physics, or mathematics. You can actually, whatever you choose, you can try to tailor yourself when you do higher education towards the need of ISRO, and they will be happy to try and recruit if you have the passion and the talent enough. I hope it answers your question.
Thank you very much.
Thank you. Yes.
Yeah, I think it answered. Now, Christy, now she has asked, it is a common belief that once you hurt a snake, it will definitely come back for revenge. Does it have any scientific basis? Do snakes have greater memory?
I think, see, I don't know. I don't know whether, see, I come from a very traditional Orthodox family where I have heard a lot of snake stories and grew up. But I have never heard too many scientists actually saying that a single snake can come in for revenge, per se. I mean, because animals doesn't actually rely a lot on revenge in the way they communicate and they in their ecosystem. They're actually, most of them try to, most of their living is to find food to survive in the nature. That is a carnal fear about the predators, and also to reproduce. So all their systems and everything is actually evolved to leave this thing. So to try and balance in this one. So most of the time, if you try to look at snakes and everything, I come from a place which have lot of snakes. Okay, and there are times that I have involuntarily stamped snake, unknowingly, or when I was riding, I tried to, cross a snake's tail and something, and these were the snakes that was living in my pond, or that coming from a village. Yeah, so we are sharing the same ecosystem in a way. But I have never seen them coming, attacking anybody, because there are so many people in my family, and none of them have been bitten by a snake yet. Which makes me believe, I don't know whether this belief is true, but from my experience and from my interaction with the colleagues that works in with the reptilian biology, at least the snakes, I don't think they actually come in for revenge per se. So it's because they just want to survive, and most of the time, if you hurt them, they actually try to run away, like every other organism is.
Yes, they are much nicer than human beings.
In a way, yes, because
in that way.
Yeah, they just want to survive, and they don't destroy things. They are not intended to destroy like humans anyway.
Just venomous too.
Very nice. Very nice. Any questions? Yeah, if no more questions, I mean, a lot of,
So,
I mean, there are more questions. I mean, do we take them? One more question, last question we will take. When will we, I mean, Hrithik Manoj has a question, when will we know if it's safe enough? I understand toxicity, example, nanotubes. Do we think we can mitigate what is safe enough? That is, how much can we?
So I think the safety base is in any biological system is proportional to the dose. Understand? So if you are trying to put even unsafe things at higher dose, you actually make it toxic. Understood? Like, say, I mean, like, we cannot, even though glucose actually gives you a good thing, excess glucose or excess sugar is not good for us. Similarly, alcohol, right? I mean, what is good in lower doses, because if you look at many medicines, their base is alcohol. It's not good if you are trying to consume them in large quantities. Similarly, for carbon nanotubes and all these kind of nanomaterials, it is the same thing. So it depends upon what kind of dose we want to what kind of dose that we want to introduce them in our biological systems. So till a limit, I hope that if they are biologically compatible, till a limit that they will not restrict too much of biological pathways and too much of cellular function, but after a certain dose they will start affecting, they will start interfering with the biological pathways, which will eventually affect the cellular and organism function. And it is that dose that everyone you need to find out if you're trying to play with the carbon nanotubes or nano rods or nano motors that could modify biological function. So again, you have to remember a cellular function is a series of chemical reactions. Okay. So the more, if our dose is going to depend, is going to alter the rate of any chemical reaction, that is when we need to be worried, and we need to be worried that it is going to become toxic to that environment. I hope it answers your question.
Very nice. And maybe also, since each person is different, the dose may
Yes.
also be different for individuals. Yeah.
True. Because, like I say, we can even say, we have to, when we try to take. I don't know whether you have given medicines to small children. When you were a small child, you would drink the medicines in a small teaspoon. But when you
Yes.
become, you start drinking in large spoons, or like the large
Tablets.
things.
Yeah.
Okay. So.
Okay.
Okay, so I hope it helps. Yes.
That's great. I mean, I would like to tell one of the students to give a feedback about the talk. So maybe one of the students come forward and give a feedback. Namita, are you around?
Yes, sir. Good evening, everyone. Respected Principal, Reverend Father Dr. Prashant CMR, distinguished Chief Guest, Dr. Deepak R. Nair, Dr. Deepak Nair, Head of Department, Physics, Dr. Program Coordinator, Dr. Sunita. So this program was basically organized for first year science students. It was really inspiring, sir. And we had an insight of how science links with our everyday life. So like it's a natural phenomenon. Every day people come and ring bell. So what how the process happens in the body. It was really an inspiring section. So we started with a talk on high resolution microscopy and nanoscale biology. Then, which was organized for first year students. So after that, we moved on to nanoscale, that is reducing the dimensions to a size of 1 to 100 nanometer. Then we went to nanobiology, and you also discussed about the help that nanobiology does in the field of diseases like sickle cell anemia or maybe Alzheimer's. Then we went on to super resolution microscopy, in which we understand things at a level of molecules. Then after that, about the synapse and neurons and all the processes that just happen in our everyday life. Then we went on how the brain processes our information. So basically, information processing, the whole process. So I'm thankful you explained it in a nice way. Then we also saw that how to link important things with the subject that we study. So basically, we are all science students. So the processes that happen in our life, we can link it with the subject that we are studying, and what to pursue after our BSc course. So in which field to go and what all you can become. So that was really inspiring, sir. And now I'm thankful to Jimmy sir for giving us this wonderful and rare opportunity to interact with such an eminent personality. And I also thank Deepak sir for spending your valuable time and interacting with us. Thank you.
All thanks to Deepak, sir. Not me, okay, don't thank me. Okay, now anyone from outside Sacred Heart College? Thank you very much, Namita, and for that wonderful feedback. Anyone from outside Sacred Heart College who would like to give a feedback? Sacred Heart Collegeന്റെ പുറത്തുനിന്നുള്ള ആരെങ്കിലും ഉണ്ടെങ്കിൽ അവർക്ക് ഫീഡ്ബാക്ക് തരാം. maybe Pawar Komal, who had asked the question. Pawar Komal or Pawar Komal. Okay, Kirtika R.
Yes, sir.
Yeah, I mean, maybe you give a feedback. How did you like it? How was the talk? Pawar
Yes,
Komal. Maybe you introduce yourself.
he gives very nice explanation, which helps us to understand many things. So it is very nice meeting, and I like to attend that meeting next time also. So thank you, sir, you and also Deepak sir, and so your institution. So thank you, sir.
Okay, Pawar Komal, where are you from?
Maharashtra from Pune.
Okay, nice. Very nice city. Okay,
Thank
you very much, Pawar Komal. Now, is there anyone else who would like to give a feedback? One more feedback? Anyone outside Sacred Heart College, please come forward. And if you liked it, we will have a series of such talks. If you liked it, please come forward and give a feedback. Aquilin Madonna, I see that name there. Hello. Or okay, let's then maybe, as always, some of the students are a bit shy. But you know, overall, it was a wonderful session. Yeah, I would say that it was really wonderful. And the last task is that of mine to give vote of thanks. So I should express sincere thanks from the bottom. But I would definitely say that very much thankful to you, Dr. Deepak. And I would also thank Mini and the wonderful 3 kids who have actually spared their father for us.
You have some network issue, Imisa.
Much. Thank the audible now, more clear.
Now it is okay, but there was some problem earlier. I think you have some network issue.
I think it's okay. Yeah, Jimmy is back, I think.
I think anyway, thank you very much, Dr. Deepak.
Yeah.
Nice talk, very informative.
Thank you.
I think all the students really enjoyed and motivated and inspired.
That's great to know. I hope some of them really becomes, someone really whom we can also look up to.
Yeah. Correct. Because you have got students from all over Kerala, even outside Kerala.
Okay.
All new entrants to the scientific world.
Well, it's a very fantastic initiative, you know. I mean, we generally don't have so much such initiatives in other places in India, and it is fantastic to know that, you know, you guys are organizing it. And I hope the students actually take a full, full, what would I say, full entertainment. I don't know. They really get both entertained and informed. I hope.
Yeah, actually, Jimmy sir is the class in charge of present BSc Physics. He is
Okay.
so that link he started this idea of giving motivation to his students, and that turns out to be something on a broader perspective. He planned to extend this talk to others also.
Okay. Okay. That's very nice. Very nice of you.
Okay.
So yeah, if I think if we are done, I don't know. I think Jimmy I think he's probably might be having difficulty to connect. Huh?
Yeah,
So
yeah.
I
Maybe
don't. Yeah. So
even
what?
if when the power supply is interrupted,
Mhm.
thing got interrupted. In my case, initially at the start, I got a power problem like this.
Okay, okay. Now, I think if it is okay, we can, if there is nothing much, I am fine with ending the session here, not a big problem. So I am really grateful to both students and you guys, all of you for organizing.
1 minute, sir. Let me call Jimmy. 1 minute. Call Jimmy.
Yes.
Shall we finish up the meeting? 1 minute. Let me check
Hello.
whether he is
I'm sorry that I went.
okay,
There was
okay.
an issue, network issue, and wonderful. Thank you all. Thank you very much, all the audiences. Thank you very much for this wonderful, for coming
Jimmy.
here and making use of the occasion. Yes.
Yeah, thank you. So
Deepak, I think you might be, Dr. Deepak, you might be really tired right
No, no.
now.
It's
So I
fine.
think you are also waiting.
I think if everything is fine, I was telling my thanks to everyone, and if everything is okay, I can log out, right? Hello? Jimmy, Dr. Sebastian?
With your activities.
Yeah, yeah. Okay, okay. Yeah.
Okay, I will call you. Thank you.
Okay,
Thank you
okay.
for accepting.
My pleasure, my pleasure. Right.
Okay, da.
Sir, it was a nice session.
There was a net. Okay, thanks, Miss.
Okay, but still, yeah, it was a beautiful session. Yeah.
Thank you very much, Miss, and thank you very much, Sibi sir, and thank you all. Maybe we call it a day. We will stop for today, right?
Yeah, yeah, sure, sir. Yeah, sure. Yeah. Okay.
Sibi sir, shall we stop? Muted. You are muted.
Okay, we will stop now. If any participant has got any queries or they want to get certificate,
Yes.
I think.
Yes. Sibi.
Okay, if you have anything to ask,
Thank you
you
all.
can ask Dr. Jimmy. Any doubt, clarifications, etc. Okay, thank you.
Thank you all. Thank you, dear students.
Session.
Thank you all.